Good afternoon, students. And today's experiment, I'm going to explain you how to do sequence analysis by using alignment method. Uh, in this, there are two types. One is blast, and the other is multiple sequence alignment. First of all, you should know what is meant by sequence alignment. So alignment means we are going to check that how much amount of DNA or RNA or the protein sequence is matched with already deposited sequences. So we are going to find the match, whether it is matched or not. If we get the high numerical results, means that match is good. Okay, like that. So we are going to find the homologous sequence, identical sequence, similarity, how similar it is. So you can use DNA sequence, RNA sequence, or protein sequence as a query, and you can run the query against the proteins or nucleotides deposited in the database. So that is the sequence analysis introduction, and here aim. So what is the aim? You are going to do the blast and MSA, and what is that? Identify the homologous sequences. Suppose you have a new sequence means you can easily identify that is there any related homologous sequences are already in the database. So that is in that point of view, the blast can be used. Both sequence alignment can be used, and determine the sequence similarities, how much it is similar, and what are the conserved regions present in that. So we can find that, and the analyze mutations. Mostly the blast sequence alignment is used to find the mutations, to study the mutation and variations within the sequences. Okay, so the results in the results you can find a slash in between the sequence letters. You can find a slash, hyphen like structure, star, colon, semicolon, and those things. Those are all indicating the mutations or replacements. Right. So many type of replacements also you can find that in the environment. Okay, don't worry about this thing. I will show you when I get the result, when I explain the doing the result. Okay, introduction. So basic local alignment you are going to do, and multiple sequence alignment. Everything is a story. Then applications. So genomic research. So you can easily identify the genes, mutations, conserved motifs in the sequence, both in the DNA and protein sequences. And here the next one is about evolutionary biology. By producing the result of BLAST and MSA, you can alignment, you can use the result to create an phylogenetic tree. Um, from the tree, you can find a relationship between the species. Okay. In drug discovery, obviously it is using for drug discovery to find the active site and the drug properties. Suppose if I have a new sequence, that sequence is nearly 80 to 90% of the sequence is matched with the already available protein means. So we can use the same drug for that. We can test with the same drug how it is behaving, is it inhibiting or not. Maybe use the uh, is that produce the same amount of action or not. So we can easily find that. And vaccine development, you know that already we are using RNA vaccines, DNA vaccines nowadays. Should be aligned and get analyzed by using this method. Okay. And disease diagnosis, nowadays we are using so many biosensors embedded with biomarkers. So we can use that detecting for genetic mutation and associated diseases, right? So we can use that PCR also. So PCR we can amplify and we can align with the to find a mutation. If mutation is there means there will be a chance for disease, right? So protein function predictions by identifying the functional domains and active sites of the protein. So in forensic science, we can isolate the DNA from the samples such as nail, hair, flesh, skin, sperm, and milk, other body fluids. Uh, so we can easily find that cells in that, and we can isolate the DNA. We can compare the DNA of criminals. We can find that. Okay, this is a way. The classical example of application of alignment is only the one is to find the family of the uh, sample. query, which means phylogenetic tree analysis. The second one is in forensic science. Okay, the protocol about this BLAST is you can access the BLAST and the server, and you can choose that whether you are going to do an nucleotide blast. Nucleotide blast means the query should be an nucleotide sequence, and you also choose for the protein blast the query should be a protein sequence. So in your case, you should do both the uh, queries, right? First complete the nucleotide and next go for protein and show the result with the discussion. After choosing that blast A and blast B, you can go for both cannot be done in simultaneous. Right? You should first go for nucleotide and next you can go for protein. Okay, after entering the sequence, you can go choose the databases, which databases you are going to compare with the query sequences. Okay, mostly non-redundancy is used. So NR for protein sequence and reference for RNA sequences. And the other suitable databases or repositories means suppose if you are using a 16 RNA sequences means there will be an option available with the strains. So in that I will show you in the future experiments. Right? Okay. Don't worry. Then after that everything is done, you can just click a blast, this will run and it will produce the result. So in the results you should make how much percentage of identity is highly shown. So you should choose the high percentage identity with the very lowest E value. The lowest E value means the uh, good result. So the largest negative value means the good result. Okay. So by this way, you can find that the similar sequences, maybe in a protein or in a DNA sequences or RNA sequences. After that, you can download the sequences in a FASTA format. This result is used to do a multiple sequence alignment. This result also used to create a phylogenetic tree, which is your next experiment, I think. Okay, this is about protocol. I will just go to the practical. You can go for blast. You just type the blast in that. You can go for that website. Simultaneously, I will show you. I don't have sequence. You for your case, you have sequence. I need a sequence to do that result, do that blast. So homo, no, what is that? Hemoglobin. I will do hemoglobin. I need a protein sequence though. For that I have used Uniprot. Suppose if you need a nucleotide sequence, you can go for NCBI. In NCBI also you can get both protein and this nucleotide sequence. So I will choose, for example, I will choose this one. I will get the sequence here and I can download that FASTA format. And this is the sequence of hemoglobin. And now you can download for this. This is the page of Blast, official website of Blast. So see that you can find nucleotide blast here and this side protein blast, and you also have special thing. This is translate a nucleotide with a protein and a protein with nucleotide. So you can use whatever you want according to the need. Okay. Um, First you have to choose nucleotide blast and submit the gene to the query box. For this practical purpose, I will use the protein blast. So you can do the same by using that one nucleotide. Okay, protein blast. So you can paste here, or otherwise you can open the file which you have already saved in the FASTA format. Right, you can upload this by using the choose file, and here I will paste because I don't have any file, so we can just paste it, copy and paste here. I have just pasted here, and you can go for job title. In this is automatically retrieve the job title from the file. In your case, you can give type here. You can type unless if you left this box for free means what happens? It will not run. So you should type anything. And here databases, experiment databases. Mostly you can use standard databases. It will vary depending upon the sequence you are giving. If you are using a nucleotide sequence means it will show other options here. Okay, so databases mostly non-redundantal. So redundancy means error will be less here. So you can use, if you want to more stringent means you can use PDB also, protein data bank also. You can get very good results, but your sequence will be compared with the protein data bank sources. Okay, usually you can use non-redundant protein sequences, and then after that you can check the algorithm. So what kind of, this is the protein sequence for analysis. So proteins blast is defaultly selected. You can find here also. Suppose if we are working in nucleotide sequence means it will be on, right? That is blast n, and this is blast p because I have given the amino acid sequence. Okay, and now you can use that. So if you want to do specifically means this is uh, position specific iterated blast. So we can search the position. We can search it. Iteration means nothing but the computational computer word. You can like a loop. It will go again search. It will search. If it is no means again it will go search like that. Okay, you can hope you will know during the theory classes. This is, if you want pattern iterations means you can click here, and sometimes you can go for which means blossom, palm like that. Those also available in that. So those are our algorithm. See that? See? No, it's not here. Algorithm parameters. Ah, see that? Scoring matrix. Right? Blossom 62. Uh, while you are doing, you just leave it as default, right? And I'm just saying you should know that what is going in the background. So these are the matrix types, right? Blossom 62. You should know what is mean blossom. I think this is substitutional matrix, block substitution matrix, and you can also find PAM 930 point accepted mutation. Hope you will come across these terms in the theory bioinformatics classes. So these are things in that. You just minimize here and just click the blast. Now you can get the result within a few minutes. It may take 5 to 6 minutes to produce the result. It will compare with the available sequences. So I have given hemoglobin subunit beta, right? So we have to wait until the results produced. Maybe it gets more time, which means it gets more hits. It gets more similar proteins, and it depends upon the internet connections also. It is still searching. Oh my god, what happened? Sometimes it may took time like that. Up to 27. It takes more time. I should change the protein sequence. What happens? So still it is running. It came at last. Okay. And here you should find that E value. See that negative, no negative. And we should concentrate on personal identity. It should be at least high among them, right? You for your sequence may not get the 100% sometimes. So you should choose which is the highest one. So you can list by using the tab. See that all hundreds comes together. So you can choose that and then you can check E value is negative. This is more negative, I think. Query coverage also 100%. You can choose that. To see that graphical summary of that, you should take the snapshot of both um, this description and you can explain what is the percentage identity you have taken and the e-values and query coverage. And this graphical summary, you should denote this color identifications. For our sequence, in this example sequence, there is no other color, only red is here. So red meant by more than 20, more than or equal to 20, 200, 200, which is highly confidence. So you can believe that. Okay. And here alignment score, the 200 means alignment score. It is highly aligned with the other proteins, right? Alignments, you can see that there are 147 amino acids given. There are matches one, like that you can find so many matches here. It will goes on 100 sequences. First 100 sequences is shown here, and you see that there will be a query. This is our own protein query, amino acids 1 to 60, and then 61 to 120 here, and 120 to 147 here. Okay, and this is the subject one, this species, and this is the result, whether it is same or not. You can sometimes you can find any space or any other symbols or different amino acids in that. These are all single letter code, right? I hope you knew that single letter amino acid code. So you can see that and find is there any gap or anything. So you can make a snapshot of this sequence result and discuss it. So I can show you any other. Hope there is no gap identified. See that there is a plus symbol in between that. That plus symbol means the amino acids, polar amino acids replaces polar amino acids. Otherwise, negative amino acids, non-polar amino acids get replaces with non-polar amino acids, which means there is no that much functional uh, differences between the proteins. Okay, so this kind of sometimes you may get slash, sometimes you may get a hyphen, sometimes you may get colon structures in that. You should know respected what it denotes respectively, right? So and here is the substitution. Sometimes you may get deletion also. That and here in this example nothing is here, everything is okay. There is no that much deletions are found, deletions or insertion found. Okay, anyway, this is the last result, alignment result. Then you can find the taxonomy of that. Say that is the what is that route I have taken. Somewhat amino acid, right? Hemoglobin amino acids. And the other sequences, synthetic construct also available. Human open reading frame, gateway, entry vectors is Homo sapiens, primates is available. So by this way you can easily find that taxonomy in that lineage organism also hemoglobin beta partial, right? Partial sequences has been submitted. So that is this is the result of that. And the taxonomy, see that artificial sequence maybe in a construct and synthetic construct and ORF gateway entry vectors. So by this way, you can find which proteins it is or which is related to the sequence are related to which protein or which species. Okay, now you can go for descriptions. After descriptions, you just this is the next level of blast. You can just create a tree. For the namesake, now you create a tree. We will see that how to create a tree in a separate experiment also. This is how phylogenetic trees are created by using blast results, right? So you can just copy and paste that. You can just download this. Download this as a PDF. It will defaultly download this. You can find options anywhere here. And few days ago, I have downloaded just I'm sourcing. So you, if you have, no, you should where it is. Move on it. Sequence label, distance, okay, maximum resolution. Where is the option for downloading? Tools. We can download it here, right? We can download as PDF file, and you can uh, cut it, which means crop it, and you can use as a what is that result. Okay. Before that, if you want to download a phylogenetic tree, means better you can choose the highest one up to 100, right? You can deselect this. You can select the highest percentage identity proteins. In this case, protein, you also do for nucleotides, right? So you go for top 10, that is enough. So now you can create a distance tree, so it will come. And here multiple alignment also we can do by using this. In the next class, I will tell you how to use the multiple sequence alignment to see that there is everything there is no there is available. Everything is a hemoglobin. So for your protein, you may get a different phylogenetic tree with a root, branches, nodes, and leaves. Okay, so this example you can download that. Okay, this is about blast. This is how you can select the more similar homologous sequences from the blast. Thank you. In the next class, we will see about